Hi everyone, this is Harrison Chenot again and Dr. Jayakumar Rajadas, the director and team leader of the Biomaterials and Advanced Drug Delivery Laboratory at the Stanford University School of Medicine. We are going to be doing the second installment of our interview that we started on Sunday. I want to give him another warm welcome on behalf of all Lyme patients and
Hello.
on behalf of all the Lyme support groups. And to start out with, I'm going to summarize what we discussed last time. Basically, Dr. Rajas told us that he's originally from Chennai, India, that he did his PhD at the Indian Institute of Technology, that he became a faculty member there for 15 years, that you developed therapy for wound healing and heart disease. You then started working with a compound known as alpha B crystalline, which is a heat shock protein, which you discovered had great efficacy against neuroinflammation. In 2007, you moved from India to Stanford University. You were originally in the neurology department, but they realized how useful you were in commercial drug development, that you then started working on therapeutic agents for Alzheimer's disease as well as neuroinflammation. You have about 72 patents, 29 to 30 of them are in commercial development. The studies that you've shown on disulfiram show that it reverses pathological processes, anti-inflammatory and antibacterial effects, that it somehow is able to stop the upregulation of pathological proteins. The way you started in Lyme disease was in 2011. You began working with the Lyme Working Group. You met the founder of the Bay Area Lyme Foundation. She gave you great support for 4 years. And it was in 2015 that you discovered disulfiram. In 2016, you published a paper. So far, your studies are showing that disulfiram is not very stable by itself. You need the intact molecule to really kill Borrelia very effectively. You also said that it's best to delay the absorption to the lower intestine. That allows for superior eradication of Borrelia, and that the prodrug in the case of treating Lyme disease is different from the prodrug in the case of alcohol abuse issues, right? So
That's correct, yes.
you have different active molecules depending on what your use case is. In this case, if it's Lyme, you actually want the molecule exactly the way it comes to be administered throughout the body. There are 4 or 5 metabolites that you know of. You would like to research that further, but you're not exactly sure how important of a role they play. You've found that disulfiram binds to 3 outer coat proteins, which allows it to get into the Borrelia, but you don't know exactly how it kills the Borrelia once it gets inside of the cytosol. You're not exactly sure of the efficacy of disulfiram against biofilms. You do not believe that resistance will be an issue with Borrelia or Babesia and disulfiram. And then lastly, you're working on a stabilized matrix of disulfiram in that you're trying to avoid the fast absorption and you're avoiding first pass metabolism, and you want slow delivery without breaking down of the drug. Is that a fair assessment of most of the things we discussed in the first call?
Yeah, it's perfect. Thank you.
I wanted to resume some of the questions that we didn't get to last time. And I also wanted to give you a chance to explain the competency of the lab and your ability as the head of the lab. Can you describe exactly what about your background and experience you believe enables you to have a unique understanding of what is required to develop a new drug for the treatment of Lyme?
So, for any drug development, we think you need two basic qualities. One is you need a necessary skill to do the uh, drug development, and also you need a passion to do the drug development because the drive to find out the cure, basically. I'm very fortunate to have colleagues in my lab, Dr. Ravindra Pothineni, and originally my former colleague, his name is Dr. Dhananjay Wang. Both of them, they started the high throughput screening in 2011 when we decided to find out a Borrelia killing agent. As I mentioned earlier, we started from the beginning. We never assumed anything. We never wanted to find out this kind of antibiotics. It's not a directed screen. It just everything from the beginning without assuming anything. We wanted to try everything possible. The reason for that is we wanted to find out molecules. We wanted to find out drug entities that ultimately totally eradicate the persisters. By the time persisters idea started coming up, so we thought we will get the best, some 20 molecules that is likely, that is, we demonstrate that it kills very well in the dish, then take that 20 and then try to screen one by one against persister and then try to find out which one clearing up the persisters effectively. In that case, we found out some few interesting molecules which predominantly clears the persisters. We always compared it with doxycycline, and these molecules are much better when compared to doxycycline. And that one, surprisingly, we found a non-antibiotic category, disulfiram. Then we focused on azulocillin and disulfiram. And as you all know, disulfiram is our kind of favorite. And we are also putting a lot of efforts in developing azulocillin for an acute borreliasis and disulfiram for basically a persistent borreliasis.
Azulocillin is the second drug that you're working on, right?
That's correct.
You've mentioned the azulocillin discovery, which is the other candidate you found being effective against Lyme disease in the acute form. Can you tell us about the other two antibiotics that are in the same class, such as meslocillin and piperacillin? Because azulocillin is very difficult to obtain, but the other antibiotics are available now.
Unfortunately, if I tell the list of the antibiotics, people can take it as a clinical advice. If they send me an email and they call me, connect me to the clinician, I can advise on that.
Is there a role for azulocillin to be used in conjunction with disulfiram if there is neuropathy involved?
Yes. In fact, the combination is likely to be better.
How do you think your training and experience is different from other Lyme researchers in the area?
Mainly in this area, most of my people who are working with me, my colleagues, they are all, some of them are microbiologists, a trained microbiologists. They understand Borrelia actually much better than our group because they have, they have a lot of training in it. And some of them are animal model people, like they developed animal models to check how the Borrelia cause disease in this animal model. In our case, we have a kind of niche advantage by training. I'm a chemist, a formulation chemist, drug delivery person. So I understand drugs little different way when compared to my colleagues. But at the same time, I have the disadvantage of not understanding the Borrelia, the persisting Borrelia, and also the Borrelia in different forms, how they survive, what are the mechanism, how they changes their, they undergo a phenotype switching, how exactly the phenotype switching happens. This kind of information we are still trying to teach ourselves. We are not expert in those areas. But we are expert. We are little having better advantage in understanding, for example, drug delivery, how to deliver the drug effectively in the gut, in the blood, in various way how to deliver it. And we also understand drug toxicity very well. We understand how exactly liver metabolizes the drug. And we also good in bioanalytical techniques. So we have a few mass specs and HPLC in our lab. So we understand how exactly this drug survive in the body fluid and how much it is delivered into the site where Borrelia is, accumulated. Borrelia, as you know, it is always in the extracellular matrix. It's not like it is there in the blood. It's, it is in the extracellular matrix of tissues, not exactly in the blood. So we try to develop drugs that will have a very good tissue penetration, distribution, And we also understand how Borrelia interacts with the extracellular matrices. So and also we wanted to translate that idea into that drug, how it interacts with the extracellular matrix, at the same time how it is delivered to the Borrelia. Let us say methylene blue we develop as a drug for Borrelia, let us say. And the main problem we have with that is methylene blue is basically a dye. And this dye got a higher binding affinity to the matrix because it's a dye. Most of the dyes, as you know, it binds to the matrix. That's the purpose of dye. You know, we like various cloth and even our skin, we take a dye and it gets stained, it never comes out. So if you are using a dye as a drug, they get distributed all over the body and they literally fixed in the matrix. So it's very difficult for them to get delivered to the organism which is there. You need a kind of available therapeutic molecule, not a fixed molecule. The molecule need to detach itself from the extracellular matrix and get into the organism so that the organism can take it. And also, you need a very good organism penetration of this molecule. So that's why the disulfiram, I always used to tell that disulfiram is kind of designed for Borrelia. So far, we think, especially the prodrug form, we see a very good affinity of we observe very good affinity of the prodrug, the intact form of Borrelia to the intact form of disulfiram to the Borrelia. And once it is bound to that, by unknown mechanism, it is getting transformed to the cytosol of the Borrelia cells. And then there, perhaps it just getting degraded, we don't know. But whatever the form it is depleting something that is very important for persisting bacterial population. We are still studying it, how exactly disulfiram is interacting with the cytosolic proteins in the Borrelia. We are still doing a lot of screen, in silico screen. We are working with collaborators to try to find out its binding partner so that we can understand and develop better molecule than disulfiram. Maybe at one point we will understand it better and we can design a better molecule than disulfiram to clear the Borrelia very effectively with literally a zero side effects.
So it sounds like your laboratory has quite a lot of capabilities. You mentioned mass spec, HPLC. You can
Yes,
do.
so we understand PK, we understand PD, pharmacodynamics, pharmacokinetics, and we measure very accurately how much drug at various after delivering to the drug, both in animals, and also we have the capability to study human drug concentration also in that human blood. We have the capability to do that. We also understand how the drug, how much it is available to be delivered to the Borrelia, because we understand in terms of matrix drug interaction, because Borrelia is always hiding in the matrix. So we try to understand how exactly Borrelia is interacting with the matrix, as I mentioned earlier, and also how the drug is interacting with the matrix, how available after this interaction or after this binding happens. So those kind of very intricate detail we try to understand and then try to develop a delivery and also a formulation that can address these issues.
When you said the word matrix there, were you talking about the cellular matrix?
Extracellular matrix, you have a cell and then surrounding that cell, you also have a collagen, hyaluronic acid, sulfated carbohydrates. A lot of them are there. So that is called an extracellular matrix.
Isn't part of what makes Borrelia and Babesia really difficult, and I think, sorry, it's more Bartonella, maybe not so much Babesia, but It's the actually it might be all three of them that they go inside of the cells. Isn't that part of what makes them difficult to treat?
We don't have so far. We don't have a lot of evidence, but there are people think Borrelia is an intracellular organism. But we consider whatever the existing understanding we have, the live Borrelia always we find in the extracellular matrix, especially when we are checking in the animal model work, we see them in the extracellular matrix. In our experience, we have never seen them intracellularly. They are not getting into the cells. It is similar, it is different from, like, for example, malarial parasites. You see all of them in the intracellular. Within the cells, they learn how to live and survive. In Borrelia, it is not like that. I mean, we think like that. Maybe somebody will come up with more evidence to show it's an intracellular pathogen. In our observation so far, we never saw anything inside the cells when we are checking in our animal models and all. I'm not, concluding anything. It is just an observation and also based on some other work other people published. But in the extracellular matrix, we also find it is not just they are sticking to the extracellular matrix. They are also sticking to the some of the cells. That is in the outer membrane of the cell. Other mammalian cells, they have a good affinity. They binds to that one. That we showed. We have data to show that it is kind of riding on the cells. When the cells moves, it also moves along with that. We have some data to show that. We have never seen whether it gets inside the cells. Of course, Borrelia can drill through the cells, and it can also drill through the extracellular matrix that other people showed. But the living inside the cell, it needs a special mechanism to do that. We are not sure whether Borrelia is having that mechanism to do that. Coming back to your question, what makes killing Borrelia is difficult? It is because it is in the extracellular matrix it is difficult. Why? Because cells, let us say that it's an intracellular organism. You can treat some drugs which is known to have very good delivery mechanism to get into the mammalian cells. So you can just treat with that. And most of the mammalian cells, literally, they have access to the blood circulation so the drug get to the blood, and then through the endothelium, the drugs can diffuse to the cells, and the cells can take the drug, and then whatever the intracellular organism it can be exposed to those drug and then get killed. In the way, let us say that, we take oxygen is delivered to all the cells in our body. That means that drug diffusion to the cells is much better than drug diffusion to the extracellular matrix. Because matrix is not having good endothelial ciliation, blood vessel formation towards it. Because generally blood vessels are forming the places where the mammalian cells are there. In fact, certain cells, they have plenty of blood vessels accessibility. Certain cells, they have least blood vessel accessibility. Unfortunately, the places where Borrelia is there, not a very vascularized tissues, it is, hiding. It may be, in some cases, it's just there where there is not much accessibility blood vessels. So that means it is like, some antisocial elements hiding into a place where there is no accessibility of traffic. So once they are there, how to find them? That's very difficult. Then how Borrelia survive? Because still it needs a nutrition need. It needs that. We think Borrelia got a very special way of making the blood vessels, which even it is a distance blood vessels, whenever they need something, to make them to perfuse the nutrient for them to have their nutritional need. When that happens, only we think it causes a lot of pain and also local inflammation around the place where it is signaling the blood vessels, that blood vessels perfuse and drip the nutrients to the Borrelia. These molecules, which is effectively killing Borrelia, they need to be having the least interaction with the extracellular matrix. It should not get fixed with the extracellular matrix, and it needs to have good affinity to the outer coat membrane of the Borrelia, and it also needs to be hydrophobic. It gets shuttled to the interior side of the Borrelial cell. All these things it needs to have 1, 2, 3. And we think Disulfiram qualify everything. It is hydrophobic. It binds well to the Borrelial membrane. That's what my collaborators think, because of specific reasons. And there is a very good interaction of the drug to the membrane proteins, and then the membrane protein also pass it on to the cytosolic membrane. For example, one of the persister model assume once it gets into a persister mode, what we call it as a sleep mode, in that phenotype form, its metabolism is kind of very, very slow. That means it just shut down all the basically drug trafficking. So only the drug metabolically active molecule, you can kill them easily because it is open for molecule to diffuse inside and then get killed. But if the molecule is shutting down all its metabolism, it's kind of within quote hibernating. It's very difficult to reach them because they don't need that much. If the molecules are interacting to the outer coat membrane and then they have a very good affinity, it is likely to this molecule, in this case disulfiram, have a better compartmentalization of Borrelial membrane, and once it get enriched even by simple diffusion, it gets into a persister very effectively. So that's the reason we think a prodrug form is much more effectively eradicating the Borrelia if it is properly delivered.
It sounds like you said that the Borrelia has a way of drawing nutrition to it, even though it might not otherwise get it. It can signal for more blood to a certain area. Is that true?
That's correct. Again, the persister Borrelia, they may not have that mechanism because there is not much nutritional need or demand for Borrelia in that state. In that case, the drug need to diffuse out without any mechanistic opening of the endothelium nearby, and the diffused drug get into the Borrelia, and then it need to get into the Borrelial membrane, and then perhaps it is eradicating it.
What is the nutrition that Borrelia needs to survive? Or is it's anaerobic, isn't it? So it doesn't like oxygen.
I don't know exactly its oxygen need. Perhaps we never studied that actually. But of course, it's like, it's a slow growing bacteria. Its nutritional need is not that high when compared to other bacteria. It divides very slow pace and it grows very slow. Like any other bacteria, it needs metal. For example, it thrives on manganese instead of iron and other heavy metal. It has a mechanism. Of course, it needs some iron also. It needs other metal ions, and it needs other nutritional molecule which other bacteria utilizes. Specifically, as I mentioned in the earlier discussion, we found out the Borrelia needs a lot of manganese traffic for it to survive. That is why we developed a molecule that can block the manganese transfer to the Borrelia. So we focused on developing a molecule that binds with manganese transporter of Borrelia, and then we found the approved molecule that can block the manganese transport, and we do saw a little moderate efficacy of killing Borrelia using this molecule to develop a better molecule that can specifically block the manganese transported much efficaciously so that we can develop a better drug to kill the Borrelia. We found disulfiram, we found azelaisulin. We thought we will focus on these two molecules, and these two molecules are easy and probably it will be available for people to use it because of the grave need. There's nothing available to treat people. So we thought, okay, these two molecules are already FDA approved. And why not we just develop this so that people will be benefited ultimately. Yeah, that's our focus. That is a unmet medical condition also.
Is there any reason in particular that Borrelia seems to love the nerve and joint tissues more than other areas, you think?
In fact, we published a paper. We just took some triacylated lipid that is basically a part of most of the outer coat proteins. And then we saw the interaction of this lipid to the nerve tissue, neural tissue, and then try to see how exactly it affects that. When we screened the borrelial protein to the neuro tissue, so far, maybe ultimately we will find out something. It appears it's not like, Borrelia got a special affinity for nerve tissue and all. But at the same time, the borrelial products or borrelial outer coat proteins, the lipids that comes out of Borrelia and lipid encapsulating with a lot of borrelial protein, those entities interact with nerves much effectively. The reason for that, number one, they diffuse very well because they are smaller entity. They can, the diffusion is basically inversely proportional to the molecular weight. Bigger entity, it diffuses very slow, and if it is a smaller entity, it diffuses very fast. So it's a small lipidated particle. They are likely to diffuse very fast out of the Borrelia, and then it can interact. And why nerve tissue, like nerve endings and also the neuro tissue got a higher affinity for these lipids? Because they are basically, nerve is more lipid. In fact, our brain have more lipid than other tissues in our, other than adipose tissue, they have more lipidic component. So anything lipid loving always get into the brain very effectively, very quickly. That's why we think disulfiram is also having a very good blood-brain penetration because it's kind of lipid-loving molecule, so it just get into there. So same way, these lipids that comes out of Borrelia, because I think I already mentioned Borrelia also, we saw shed out lot of lipidated particle when it moves. Even when it is, staying in a place, there is a lot of lipidated particle that comes out of it. Those particles can move and interact with the nerve ending and then cause a kind of signaling changes that can lead to alter different signaling mechanisms, and that can lead to a disease. We already showed in our Journal of Neuroscience paper how it exactly happens. Again, we need to do a lot of experiments to show what are the particles that kind of emanate from the live and dead Borrelia and how those particles survive for very long time in the brain and cause the disease. Because as you know, when the particle, like even the, let us say, people are having kind of after antibiotic treatment, they produce a lot of dead Borrelia, and then borrelial particles they generate, and those borrelial particles are cleared by macrophages in the peripheral tissue, and in the brain there is a type of cells called microglia, and the microglia also cleans up. They are all scavenging cells. The scavenging cells effectively clears them. But what we found out when the scavenging cells, they are exposed to this borrelial product, they produce a lot of cytokines, inflammatory cytokine, and those cytokines can cause it, pain and also trigger a disease, an inflammatory disease. So that is also another challenging problem for us when we are developing borreliocidal compounds, because there's a difference between bacteriostatic agents and also bactericidal agents. The problem with the bactericidal agents, when you treat with these molecules, let us say in our case it's Borrelia. The Borrelia takes those molecules and then immediately kind of it degrades into pieces. When it gets degrades into pieces, you are generating a lot of toxic product. The outer coat membrane, lipid components, everything form like a small vesicles and they travel. You imagine you are kind of bombing some small organism, and the organism shatters into small pieces, and those pieces form a little droplets carrying inflammatory protein, and that protein can cause pain, and also maybe that protein can that particles can trigger a production of proteins that can sustain a disease for a very long time. We wanted to avoid that. We wanted to develop something that will not make the bacteria to shatter, and that can cause a secondary reaction. If you treat something, then it just kind of mummify the Borrelia without shattering into small pieces, and then the macrophages, they come and then clear the mummified Borrelia very effectively. Whether any molecule is possible like that, we try to find out. We try to modify the existing molecule that is working very well, whether that is possible. That is the reason when we do the study, in fact, the latest paper, it is in Biomaterials, we showed we always monitor the cytokine production, because when you treat with that doxycycline at higher concentration, we do see when the Borrelia is getting cleared, when Borrelia is kind of, either they are cleared by the macrophages, the dead Borrelia, or when the Borrelia is kind of getting degraded, they always produce IL-6, a kind of inflammatory cytokines that can ultimately trigger inflammation in the body. But when we check with that Disulfiram and also, our another favorite candidate, we are seeing like less amount of cytokine production when the Borrelia is getting cleared. That's a positive note. So we always check the immune response during the therapy in this animal model. We always wanted to identify something that is not producing a lot of cytokines when they are dying. So we need to find a molecule, not only just eradicating Borrelia, but we need to make a molecule we need to identify a molecule that can literally make the Borrelia to cleared in a very, very safe and smooth way, so that we don't face after effect of the Borrelial, degradation.
So what you're saying is that when Disulfiram kills Borrelia, it does so by breaking it up into pieces, not by just mummifying it, like you said. Is that what you're saying?
What we found is there is a minimum amount of cytokine production when the disulfiram is clearing the Borrelia. That we know. In fact, we put the data in our latest publication. We are yet to demonstrate, you treat the Borrelia and how Borrelia is getting degraded in the presence of disulfiram in animals. We are yet to show that.
Did you actually analyze how the mice who were treated with disulfiram, how their behavior changed once they got treated with disulfiram? Did they get worse before they got better? or did they get better slowly?
Yeah, so let me explain this. So we have a mice model. These mice, they develop disease. In fact, you leave untreated, they develop arthritis, they develop a lot of neurological problem. They also have, as I mentioned in my last discussion, they develop a lot of pathological proteins upregulated when even in the acute exposure in these animals. It's a perfect model. But at the same time, mouse is not a small man. We are different, they are different. They're just a model. We can learn few things from them. Like, for example, we can learn whether Borrelia is cleared in mammalian body when you are delivering by orally or by intraperitoneal injection or by intravenous injection, or even when you give the drug by inhalation. We can ask those questions and we get an answer. Yes, you can clear them in mammalian body, because many drug we develop in a dish, it never kills or clears the Borrelia when we test it in animals. There are a lot of molecules when we transform those molecules with a great enthusiasm, and then when we check whether it is clears in Borrelia in animal, and we are very, very disappointed it is not clearing the Borrelia. Those are molecules, we have to drop those molecules. Fortunately, disulfiram is not like that. Fortunately, azulazolin is not like that. They are as effective as we saw in the test tube when they are clearing in animal model. How exactly it is interacting, those kind of questions we need to learn, and then we need to understand how exactly it is doing. Coming back to your question, whether how to model this animal, the behavior, like for example, the humans, when they are infected with Borrelia, different people, they have a different response. Like 15% of them, they have the symptoms for very long time. Even after antibiotic treatment, they do have the symptoms. 85% of the people, they are okay after doxycycline treatment. They are perfectly free of any symptoms. You can call them, they are cured. You don't see this kind of things in animals. Like you take 100 animals, 15% of them you don't see, like they develop a chronic disease. My colleague, Dr. Monica Embers, showed for even prolonged antibiotic treatment like doxycycline treatment in animal model, she showed in a chronic model, Borrelia is still there. This she showed. But in a human, if they have a persisting bacteria, let us say for a very long time, they have a lot of pain, they have a headache, they have other kind of problems, they develop arthritis. Whether you see a similar, exactly mirroring this response in animal model, the answer is no, unfortunately. It is animals, in our hand at least, the animal model we are using, they experience less pain or cognition problem when compared to human. And that makes a lot of challenging thing to check the efficacy of the drug. One example is we do an experiment called novel object recognition. These mice, you put a new object near to them, they try to explore it. That shows they have a very good, mental capability to check what is new. And if the animal is kind of dumb, like they have some, cognition problem, they lose that enthusiasm. So novel object recognition, we check whether, you put the novel object, how much they recognize. Like if the animal is affected by some kind of cognitive problem, like Alzheimer's disease kind of toxic protein in their brain, then they lose the ability to recognize the novel object, because they repeatedly think it's novel. So this test we do, we have some effect when animal infected with Borrelia, they lose some amount, like because of they develop a cognition impairment. And whether disulfiram is reversing or not, we are studying. We need to do like, it is not like 8 mice is enough for this kind of study. You need to use like some 20 mice, 30 mice. You infect them, and then you put the novel objects and then find out whether they have ability to recognize them. And if you have a very good effect that all the Borrelia infected animals, they lose, let us say, significantly the ability to recognize new object, then you treat with the disulfiram and try to find out whether they are gaining back their ability to recognize objects. So this is the experiments we periodically do in our lab. But again, as I mentioned, to convince people that disulfiram can reverse whatever the cognition impairment caused by the Borrelia infection, you need to put a lot of effort, resources in it to convince people. Like for example, you need to use like some 20 mice, 25 mice in both sides, and it's very expensive experiment.
As I remembered at your Lyme Mind speech in New York, you had mentioned something about the mice appearing
Yeah.
very sick, not
Yes. Yes.
behaving
Yes.
very normal.
Yes. 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 Yes.
And I was
I did experiments for that. Unfortunately, the quantifiable thing is not very significant. We need to recruit more animals to show they develop a significant change, infected, non-infected animal. And then we have to show after treating with disulfiram, the condition is reversed. So again, it needs more resources to prove that disulfiram is very effective in reversing the cognition.
But so so formally you don't have anything you can offer, but informally, just from your technique.
Informally, we do see good effect. Yes, the animals, their brain signaling changes. I mentioned this several times. After acute exposure of Borrelia, you can check their brain. We even do the PET scanning in a live animal. We scan in the way they do the cancer cell detection. So people, what they do is they just like let us say in a human, they do a PET scanning. They use one of the glucose derivative carrying some isotopes in them that can generate signal, so they can monitor that signal from outside. We feed people with that, and then it gets into the brain, and then if they have a cancer, those cells, they take a lot of glucose. So you see an intense signaling in this area, where if they have a glioblastoma, there is an intense signal in that area. The similar experiments we did, in fact, those data is there in our paper. We showed even a small outer coat protein, a lipidic component, get into the brain, how the signaling, the glucose movement, it uptake, how the brain connections, how it changes. We showed that actually. We showed in these animals, in live animals. We showed by PET scanning.
So what did the PET scan results show?
I can share the manuscript. Anybody is interested, if they send me an email. The PET scan results shows that it definitely impair for the metabolic activity of the brain, and there is a very significant reduction in the cognition network. Whatever the connection, the overall connection in the brain. So we showed that it is affecting significantly. Unfortunately, that paper is very fundamental paper where we wanted to find out an answer in case this lipids gets into the brain, the lipidated peptide get into the brain, whether it can cause a disease. That's a simple question. The answer is yes. We do saw a very, very significant changes in the brain signaling. And then we saw the cognition, the synaptic damage is happening, the synaptic proteins is altering. Even a small portion of Borrelia is causing a very big problem in this animal at very low. a reasonably low concentration. And that was kind of encouraging, but we wanted to translate it into outer coat protein particles when it get into the brain. What happens? Those experiments also we finished. We are ready to publish this work, and we wanted to do more PET scanning, and then to show. Then we wanted to take it to the live Borrelia, and then if the live Borrelia get into the brain, what happens to that? These are all the questions we are currently intensely studying in our lab. We are likely to publish it very soon. Hopefully, we will have a convincible data, like good data to show disulfiram can not only clear the Borrelia, not only make the Borrelia to die in a way it never causes a lot of inflammation to the body, but it also can, the disulfiram therapy can also reverse some of the cognition improvement caused by the acute exposure. We think we can, present the wider audience a very convincible, good data to, to prove our point. But again, we are continuously working on it. I think last time I mentioned, we have about three, three of them are working on the neuro part, and about two of them intensely working on the immune alteration related to the disulfiram therapy. And every day I always feel like data, once it is ready, it needs to be published as quickly as possible, not wait for, like a bigger, a more popular journal, but it's better to publish it so that people can get benefit out of it.
You mentioned that it sounds like Borrelia slows down the metabolism of the brain, and you've been able to observe that with PET scans. In fact, I actually did a PET scan in 2016 when I first started treating for Lyme disease after over almost 2 decades of not being treated. And when I first treated for the first time, I got very, very sick, a lot of neurological problems. And when I did that first PET scan, they found hypometabolism in the brain. So that's certainly similar to what you're describing. And I'm wondering, is that hypometabolism of the brain due to certain genes that are being turned on, or certain pathological proteins that are being activated, or what is exactly causing the brain to go into this new state?
Yeah, I have to correct you. Um, like for example, putting people in the PET scanning for different experiment, you need this kind of approval for it. Correct? It's not like anybody can walk in and then do those kind of novel diagnostic experiment. Same with animal. If we need to do animal experiment, inject Borrelia and put them in PET scanner, it needs a lot of approval. Sometimes it takes months for us to get approval to do these experiments. There is a committee, they need to check whether this experiment is needed. We have to convince them, we get an approval, and of course we need funding for it. So it's very, very serious issue. Like what all we did is we just injected the lipidated peptide that it kind of decorated the outer surface of the Borrelia. That resulted into hypometabolism, that resulted in kind of changes in the connectivity in the brain. That's all we know right now. We translated that into an outer coat protein. We have some data, we are analyzing it. Maybe at one point we can come up with a very good hypothesis. So why we get hypometabolism, why we get an obstruction in the blood flow. If they have a hypermetabolism, like hyper blood circulation in the brain, obviously they have a very good ability in terms of cognition and all. But if the blood circulation is impaired, let us say a micro kind of strokes in the brain, then the brain blood flow is impaired because blood clot all over the brain, and then the neurons and the tissue, they are not perfused well. They don't get the nutrition, they don't get oxygen. because the blood vessels is not accessible in certain area. Obviously, it leads to a kind of fog, cognition impairment. As you beautifully pointed out, it's a hypometabolism. Hypo means less, hyper means more. Whether we can reverse it, we are to do more experiments by that. There is one particular pathway leads to clogging the micro blood vessels triggered by Borrelia. Again, I am not declaring anything. We are just focusing in studying that mechanism. Because when we saw azulocillin is also kind of immune protectant, and also we also showed that it is kind of literally curing the infected animal, not just clearing the Borrelia, but it is also doing more than just clearing the Borrelia. A reduction in other kind of pathological signaling that is triggered by Borrelial infection. So when we saw that, then we indirectly wanted to find out what causes this hypometabolism, and then we found out one particular pathway that involved in the clotting and microclotting around this smaller blood vessels, and then we are now currently very carefully looking at it. Perhaps that is why people develop a lot of cognition related problem, and we don't know what exactly the reason their brain conditions are affected by borreliosis. We don't know yet. It's because of the Borrelia persisting, living there, and then they causes the disease, or it is just some kind of a perpetual inflammation triggered by a Borrelia when it was there, and then it keeps on continuing this inflammation that is causing the disease. We don't know yet.
It sounds like you're saying that there's a few possible ways it could be. It could be clogging the micro blood vessels. It could be diminishing glucose absorption. It could be causing excessive inflammatory cytokines. It could be causing autoimmune problems. There could be beta amyloid plaque. I guess some of those things are reversible. A lot of them are reversible, and some of them are not, right?
So far, from whatever the results we have with the neuro-related thing, so far, I'm not telling that's final. We think Borrelia exposure-related neuro problem is reversible. It's not like Alzheimer's disease where it just goes and then destroy the neurons and then it become kind of irreversible damage. It's not like that, fortunately.
Okay, I just want to say what hypoperfusion is in case anyone doesn't know. It basically means that the blood supply to certain parts of the brain is not adequate enough to reach a certain concentration for the disulfiram to do its benefits.
Hypoperfusion means like, for example, even oxygen supply will be impaired in those areas. Mm-hmm. A very extreme case, a stroke can lead to hypoperfusion because of the blood clot, the damages it causes to the vasculature.
Borrelia causes damage to the vasculature?
Of course, Borrelia is affecting the vasculature. We showed in our lab results, but stroke, as I mentioned, it definitely causes damages in the vasculature.
So you're saying the Borrelia damage to the vasculature in the brain is reversible, right?
I think so, based on our preclinical model. But again, we need to show how it works in human system. In fact, the clinical trial that addresses that particular issue, we specifically measure this particular issue, how post-treatment Lyme people, how their brain perfusion changes, and then how the therapy helps them. This is one of the experiments we propose in people.